Whether you keep them in your home or love to see them in theirs, these are the creatures that bring us all together.
Reptiles. We're going to be delving into the experiences of reptile lovers from around the block and around the world. This is the Reptile Talk Podcast. What's up, everybody? This is Jeremy Turgeon from Brassman Reptiles.
And I'm Rob, and I'm a Muppet that's creeping it real.
Oh my gosh.
If you're just listening to the audio, that will make no sense at all.
Yep.
Just dancing
And we just talked about not wanting to do anything crazy visual. And what is the first thing that you do is something crazy visual.
Being a weirdo.
Wow.
Yeah.
Wow.
I'm super pumped because this is kind of like a coming back to our roots and getting back to some of our first episodes.
That's right.
A year later. And I'm super pumped because we have got Sir Steve Tillis.
Stephen Tillis.
Is he Dr. Steve Tillis now?
Is he doctor yet? I don't know. Can I ever call him Dr. Steve Tillis without feeling? I mean, I'm 100% going to do it, but I'll also be like, man, I am really nothing.
Yeah. I am worthless.
Yeah. What am I? Oh man. All right, so we're talking with Steve Tillis. If you're watching on YouTube, thank you so much for jumping over, and
we appreciate you.
We definitely appreciate you guys. You're getting the back-to-back episodes because we just finished talking with Justin Kabelka for episode 52. If you're listening on just the audio, well, you can go listen to episode 52
in a week.
Or this from a week from today. Yeah, something like that. I don't know. Either way, you're gonna figure it out. Let's stop the bullshit and bring prospective Dr. Tillis.
Wait.
What?
No, do it.
Oh, okay. I thought you were saying wait.
Get to it. No, get to it.
Hi, Steve.
What's up, man?
Man, you guys overhype me too much, and don't tell me that I'm going literally back to back with Justin Kabelka. That's setting it up for me.
Next episode.
Yep. Where the hell is those ball pythons, dude? Steve's like, I have the Zio River locality normal. It's at least 6 feet.
This is not a super orange dream pied clown.
Yeah.
It's funny you say that because the only ball pythons I have are literally like either dreamsicle stuff or Zio River stuff or the weird stuff I brought back from Africa to try and prove out, and the basic morphs to prove them out with. Yeah. And one of them is an orange dream, and one of them is you name it. They know all the base genes there. They had a bucket full of them, and So yeah.
Oh man, amazing. So what, dude? How have you been? It's been a while since we've talked to you on here.
So I'm actually having a weird week, in a certain sense.
Oh no.
Yeah. So I just found out yesterday that so my first pet ball python, who I've had for 16 years, so got him when I was 11. About a month ago, he was going in a shed, and he got really uppity, and even just defensive in shed is just atypical for this snake that I've had for 16 years and used in umpteen thousand, educational presentations. And about 2 weeks later, I noticed a really big mass on the side of his neck. And then when I took him in, I scheduled a CT and brought him in for a CT, and within the week it took me to book that, it had almost increased by size by 50%. So it's not looking great for him. So, that's kind of a bummer story to lead on to, from the beginning. But the reason I'm saying that this is a weird week for me is that because of COVID, one of the safest lab activities I've been able to do is to practice cell culture. And part of that is establishing, cell lines from animals that have passed away. And one of the things that we have tried multiple times now and failed at completely is a ball python cell line. And a tumor cell is like the holy grail in terms of starting tissue from a cell culture. Because, so cancer is a cell that's figured out how to live forever.
Yeah.
So if I take a piece of that and throw it in a culture, essentially what I can do is make my first ball python immortal. So it's sad and it's neat at the same time that because of this weird, set of circumstances, I'm at a really good place in my life to be able to capitalize on this really sucky moment for me. But the significance of having this animal that, in my personal life has vastly changed the course of my life. To know that 100 years from now, this snake could still be benefiting herpetoculture is such a special feeling, and it's such a rare. Like, I could have never plot out the set of circumstances that would lead me to be able to sit in this seat and be able to do that. But it's wild.
Yeah.
Exactly. Yeah. And it's really, if that had happened 2 years ago or 2 years from now, it would have just been another dead ball python,
Yeah.
So
Yeah.
it's weird, very weird. And I mean, especially because you guys know the number of ball pythons under my care. The fact that of all the ones and I mean, granted, it is an older snake and stuff, so it would be statistically more likely to end up with something tumorous. But again, I have a lot of old snakes that I manage, and for this is the only one with a tumor. So
Yeah. That is definitely a weird week.
It's a weird week. Yep. But yeah, the prospect of an immortal ball python is what a neat legacy to be able to leave behind. Like again, just to match a personal legacy in my own life to a scientific one is God, such a unique feeling.
Yeah, I just want all of the non-ball python people to clip that immortal ball python.
Yeah, what are you gonna name that morph?
Oh gosh, what's funny is that it will get a cell line name, so
but there you go.
Probably like Pappy ball python. Yeah, nothing clever, but who knows?
Dude, you
maybe
gotta at least name it after yourself.
Yeah, something like that. But what's really funny is that like sometimes the cells of the animals kind of take on the characteristics of the original animal they came from. So like I was setting up a bunch of Galapagos tortoise cell lines over the summer, and this was like a Galapagos tortoise that died of old age. So like a very old, already giant old tortoise, and its cells were not aware that the composite being that they were a part of had decided to part this world, because man, literally every tissue I threw on a plate was just grew a cell line. It just everything
That's
wanted to. All those cells were very much keen on keeping going. So that's
insane.
Yeah.
That really is insane. Damn it, Steve.
My favorite cell culture story is so there's a corn snake heart cell line that we tried to establish, and man, it was like a month after we had taken it out of this, removed the heart tissue from this dead animal. And those heart cells were still beating in culture in the plate. It was wild. Yeah.
What?
Yeah, 30 days after, they were no longer part of a snake.
And they were still going.
Yeah.
That's insane.
What the hell?
So we're kicking this off with some insanity.
Yeah.
Right.
Yeah,
now.
going real weird places real quick. So
Jesus.
I just again I figured you guys called me up yesterday asking for podcast material, and I'll be like, well, I certainly have had it stumble across my plate in the last 3 hours, so
Holy
God.
crap.
Steve, you live a pretty awesome life.
I really do. I really can't understate it to the I have none of this planned. Like I just stumble through life and cool things happen to me, and the way I describe it is I'm a very lucky person. It's either extreme bad or extreme good, oscillates wildly, but if anything, I'm lucky, and it's the first ball python dies, but also I can turn it into an immortal cell line at the only point in my life of which I'm probably ever going to be able to do that.
Yeah. Jesus. This is
so wild.
Wow.
It's serendipitous. It's
damn. But wait, and again, Steve, I feel like nothing.
Yeah.
Again, it's you guys give me too much credit. I'm a lucky person.
Immortal cell line. I don't know what you're
I just made I'm going to make the only immortal wall python, and dude, I can't wait to tell Kevin that.
Ooh.
Be like, hey, you think you're badass, Mr. Evil Morph God? Get this. Yeah.
No, sorry, I should be careful. I don't want to jinx it because it's still a snake in my snake room. So the success rate on establishing cell lines is not great. But that being said, this kind of scenario is a type of scenario which we typically have success with. So we'll see what happens. But
Man.
my fingers are crossed. It would feel fitting, you know. I would feel like that is a satisfactorily end point at that animal's life. It's lived a full, you know, full life at that point.
Want
So.
for it.
Yeah, for sure. Man,
It's more than a lot of people can say in the reptile hobby. I'm just saying.
That's-
like,
that is very, very true.
you're like, I had my ball python for 5 years and I died. And you're like, oh no, I've had this thing for 16 years now. And it developed a tumor, which is really weird because they don't usually always do that. So
Yeah.
Yeah, and I mean, especially like again, it was like I'm talking about this ball python has a tumor that is 3 inches by about a half inch on its neck.
Holy
Like,
Oh, wow.
and that was not there a month ago.
wow.
That's crazy.
So whatever it is, it's very aggressive, and very aggressive tumors also happen to be very aggressive cell lines. So
Yeah. Wow.
Hmm.
All right. Where do we go from here?
I was gonna say, well, I've been talking to Steve about I've been watching some of the blood and short tail things he's been doing, and he's got some cool stuff going on over there. So you're feeling pretty good about the direction those girls are going in?
yeah. So I tried something different this year, and it worked really well. Yeah, kind of too well, because I had kind of anticipated a success rate that I would typically expect. Not like, so normally my success rate on blood's like 50%.
Yeah.
Mhm.
And this year it's something like I think 80%. So what I did was I fed them pretty decently over the summer, you know, nothing crazy, and then just did not feed them at all November, December, and about half of October. Then when January hit, I just fed them a jumbo rat a week for about 5 to 6 weeks. And at that point, almost every single female had ovulated.
Wow.
Like,
Damn.
females that went last year ovulated. Even in Brownger's time, which I've never had that happen, ever had a female lay back to back. So whatever it was, I don't know. And it's also been a mild winter here in Florida, so you know, maybe it was nothing to do with my feeding, but I tried something different and it worked really well this year, and I wasn't prepared for that. So and you know, there's a lot of ground to cover between here and babies on the ground, so you know, you never
That's
let the words get too far ahead of you, but we'll see.
True.
Yeah. Damn.
That's pretty exciting.
That's gonna be a lot of babies, bro.
It is, and it'll be a lot of cool stuff too, because it's like every I've refined my collection to a point where every element of it has an explicit purpose, and so every clutch is going to be a great clutch, and that's a good place to be at.
Yep.
But it's also a not a great place to be at in some senses, you know.
Like trying to find all of the mice to feed all of those babies.
Yeah, or just the time, you
The
know.
time too.
Time is my absolute rarest resource right now, and so you know, I'll have to get creative.
Yeah. Well, I mean,
But yeah,
it's not like, you know, you do anything.
yeah,
It's
right,
just,
yeah.
You know, I don't know why you're so strapped for time. Dude, I mean, just for the people that don't know, like when I text Steve about something and I get a response like that same day, there's like a little like that happens when I see his name pop up, because I'm usually genuinely like, he's not going to see this until like midnight or 1 a.m. if he's still alive at that point, and then maybe I'll get a response the next morning if he doesn't want to just leave me on red. So
yeah, no, conversing with me is really wild. You never know when it's going to get a reply. I mean,
yeah, exactly. That's why I'm like, hey, I talked to Steve and like he's ready to go, and I don't know how
this
to
right now.
Yeah, I don't know how to feel about that, because this is intense right now. Oh man.
well, I'm glad to be on. Am I your second guest?
Yes.
First double guest? Yeah.
Yes.
Sweet.
Yep. Hell yeah.
First.
Well, I'll
repeat
take that as
offender.
another little badge there or something.
There you go.
Boom.
There you go.
So you've been doing a lot of research and doing a lot of work, and then you're working on your doctorate, right?
Yeah, so I'm working on a PhD. So I'll be doing my qualifying exam in the next couple of months. After that, I will be a PhD candidate. But right now, I'm just a PhD student. So, and then 2 years after this, I will defend my dissertation and then be Dr. Tillis. So
we got time.
We got time.
Yeah, at this point I'm like pre-doctor. So like I'm like way barely in the running.
So right now it's just a little D and a little R. That's it.
Yeah,
sorry, I'm not feeling any kind of anything but sarcasm and terrible jokes. That's what's happening tonight.
You can call it a pre-HD. We'll go with that.
Pre-HD. Okay, all right.
The
I like
WebMD?
that. Yeah, the WebMD. Oh no.
He knows a lot.
Anything but the WebMD, just not the WebMD.
Not that. I'll take the pre-med MD.
So yeah, and so I've been doing my PhD research on reptile pathogens, but largely that's shaken out to be predominantly nidovirus stuff, or really serpentovirus is the more appropriate name to call these guys.
Yeah.
And
Man.
we talked like a little bit about that the last time, but there's so much that has changed in the interim, and since we spoke to you a year ago.
Yeah.
What is some of the stuff that you've seen? What is some of the stuff that's going on? That's some of the stuff that you can talk about.
Yeah. So what I've been doing as of late is both in vitro experimentation, so experimenting with virus in cell culture, and that's the kind of stuff where we can start figuring out what kind of host range these viruses might have in vitro, at least. If we throw a ball python serpentovirus on Galapagos tortoise, is it gonna grow there? Or maybe it infects, but it doesn't infect that well, so it doesn't actually grow, but we can still see that there's an interaction there. So that's the kind of questions that we're really trying to figure out with that type of research. And the other thing related to that is really a lot of the like sanitizer type studies and environmental stability. So I'm actually really excited. Tomorrow's the first pilot experiment with some of the sanitizer stuff.
Cool.
Yeah, so that'll be unique to see. I still think we need to probably tweak that assay a little bit before it's ready for full scale experimentation with it. But it'll, knock on wood, hopefully I get some decent pilot data to kind of run with that. And then I just finished some environmental stability stuff where basically we take aliquots, tiny portions of virus, and leave them out on, at room temperature in the freezer or fridge for, basically I take a sample of virus every 2 days and see how much live virus is left still. And it is actually looking pretty good in that. So right now we experimented with ball python nidovirus, ball python serpentovirus, which in my mind has earned the place as probably the serpentovirus which I would rightfully feel the most scared of. No, shoot, I forgot where I was going with that. Yeah, so we environmental stability, we found that at room temperature probably about 8 to 10 days. So that's pretty in line with what we expected off other research with coronaviruses. So good to know that the environmental longevity is really pretty limited, which is again pretty much what we expected. But it's one thing to hypothesize that based on similar viruses, and another thing to actually have here's the data that says that that's not so much the case. So yeah, and that's research that is directly funded from Carpet Python or Carpet Fest. So.
That's awesome.
Yeah, that there's your research always at work. It is happening. COVID sucks, but stuff's moving.
That's cool.
Yeah, that's really cool. Yeah. And
I want to backtrack just a little bit. If someone is listening to this podcast and has never heard of serpentovirus or nidovirus, what is like a quick rundown of what it is, what it kind of presents as or doesn't present as? And just like a quick little synopsis of like what it is that you're researching.
Sure. Yeah. So serpentoviruses are a large family, a large genus of viruses in the order Nidovirales, which is actually the same order as coronaviruses. They're not super closely related to coronaviruses, but I mean they're certainly share more in common with each other than they do to any other major type of virus. So much like coronavirus, these are predominantly respiratory oral tract type viruses that target those types of tissues. And they certainly seem to play a large role, if not a majority role, in causing respiratory infections. So the recipe for a respiratory infection is definitely not as simple as snake plus virus equals sick. But a lot of times, if you get a sick snake and you look at the recipe, a serpentovirus is in it.
Is there?
If that makes sense,
Yeah.
So if you see a virus, I don't automatically think it's sick snake. But at this point, when I see certainly a python with a respiratory infection, I mean, if you're not like, keeping it at 30 degrees or something like that, and I see a rest, if you have a room full of otherwise healthy snakes and you have one snake that is not doing as great, that's when I really start to, there's no, that one cage isn't sitting at 60 degrees or something like that. There's something different going on in that animal, and there goes that. But so a lot of there, I want to walk a fine line here, and I want to make sure I point out two things. So I will be speaking at some points here as Steve, PhD student, talking about the legitimate stuff that I can talk about with legitimate research and stuff like that. And I'm also going to be talking as Steve, the guy who's been keeping and breeding pythons for 12 years, kind of fitting what I have learned as a PhD student into what I've seen in my experience with herpetoculture. So, some of the stuff that I'm, so I'll explicitly talk about the role serpentovirus plays in my colony. And this is where I have, Steve, the snake keeper hat on, if that makes sense.
Yeah.
So what has changed in my mind? So we did a little bit more deep genetic sequencing of full genomes. And what was interesting is that we had a reticulated python that diagnostically tested positive for a virus that's actually very similar to a reticulated python in my colony. And this is a retic that I've had for a very long time, never broke with illness. I think the thing is entirely bulletproof. I've tested a lot of other samples with very similar viruses and not seen a virus or not seen illness in any of these snakes. So in my mind, that's a virus that I'm kind of a little bit less concerned about. But interestingly, we had a diagnostic sample of a sick snake that tested positive for that virus. And so we sequenced the entire genome of that, and we actually found two viruses in that snake. We found a reticulated python virus, which was actually at a pretty low level, and we had also a virus that kind of looked like a green tree python virus, which is a little bit more known to certainly cause illness. And so, kind of hypothesized that this snake had, literally two viruses, one virus in it that probably didn't matter for the health of the animal. Right. And the other one that was causing it to have respiratory infection and causing it to have a diagnostic sample sent in, so it's
wow.
So what is interesting, because when you look at the amount of sequence we generated, it's basically potentially the green tree python virus was similar virus, was at 8 times the level as the reticulated python virus. But because our initial diagnostic primers liked the retic virus more, it outcompeted it in amplification. And so that was the only virus we picked up on our diagnostic sample.
Really?
So what's interesting is we'd be able to say, we would think just based on the diagnostic sample that this retic is a retic virus and that's what's causing it to be sick. And that was probably maybe not the case for this animal. And that's not to cast doubt on any of it so much as just to say that it's really freaking complex and there's a ton of viruses out there, and sometimes they matter and sometimes they don't, and sometimes that can occur in the same animal,
That really is crazy.
Yeah.
So
with that in mind, here is how I now manage my colony. So I should start back that I had an outbreak in part of my adult blood python colony in like 2017, 18 maybe. And so as part of that, I tested every single snake in the colony and found 9 serpentoviruses, of which 2 were actually associated with any sort of illness in my colony. And at the time, I took a very heavy-handed approach. I euthanized anything that was positive, with the one exception of the reticulated python and Kenyan sand boas that were positive, because the risk that they showed to me seemed pretty minimal, especially because I don't have a bunch of sand boas or anything like that. They're literally, living in a tank in my living room kind of thing. So, that was a risk I was willing to live with. But aside from that, I was very heavy-handed and euthanized, almost everything. So that was my year 1 result. And what was left of my colony was holdbacks and neonates, which took me, a number of years to raise up, and now they're starting to come online. And so theoretically, everything in my building now has been tested negative 3 times. And then last year, I had a gravid female go through a prelay shed, break with a super mild resp. After her prelay shed, totally cleared it, but I went ahead and tested her, and she tested positive for blood python serpentovirus. So, so here's the thing. A PCR test is answering the question, did we detect RNA of this virus on the swab? And there's potentially a lot of reasons the answer to that might be no, besides the fact that the snake doesn't have any virus. It could be that the primers don't pick it up. It could be that it's at too low of a concentration. What I think is going on in my particular animals is that when these animals aren't stressed, they're just not shedding enough virus to be picked up on a test. They're not stressed animals. They can have an immune system capable of keeping an otherwise kind of inert virus for blood pythons in check. And honestly, this is why when you look around and you hear about blood pythons sensitive to RESP, like atypically, like knowing what I know now, that hits every bullshit red alarm in my brain of what a low level serpentovirus absolutely acts like. So what we don't know is if these guys are completely like intermittent shedders, or if it's just, sometimes we don't know what the sensitivity is on the test to know what the threshold is for it to pick up. And the other challenge too is it's likely to be slightly different for every virus, for the exact same reason that retic had 2 viruses and yet diagnostically only tested positive for 1. Because if you have one virus that those primers really like, it's going to be outcompeted. The other virus is going to be outcompeted by it on the test. So it's tricky. So anyway.
Man, yeah.
Last year I handled it where I'm like, I'm going to, anything that broke with that kind of RESP, I went ahead and euthanized. I was like, whatever virus is in this individual animal clearly has conditions under which it can be disease causing. And so in my mind, that is absolutely, grounds for not being in the adult colony. But this year, I'm going to try something a little different, because I think these snakes are, these females, after being gravid, after laying their clutch of eggs, after no longer being stressed, are probably I know they're going to go back being healthy because they've already shown that, but I think they have a good chance of going back to actually testing negative if that virus is brought back down to a low enough level where it's no longer really shedding it. So this year, what I'm doing is any sort of female like that. Again, I'm talking like a super mild RESP. Like you might even miss it if you're not looking for it, but I've so honed in on what I'm looking for now that, if I see any hint of it, instead of just euthanizing those animals, I decided I'm going to isolate them this year and just keep them isolated until they test negative and determine when that might be. And so here's kind of what has prompted part of my shifting in that mentality. And that was there's actually a Radiolab episode that was just put out called Dispatch 14, COVID Crystal Ball. And in this episode, they looked at immunocompromised COVID patients. And what they found is in one immunocompromised COVID patient, after each kind of attempted treatment to save this person, the virus within their body actually changed in terms of which kind of phenotypes of the virus were being represented. in which proportionality. So like after an antibody treatment, all of a sudden the UK, a virus that looks a lot like a UK variant started showing up in this. So what they found is that basically under certain sets of conditions in immunocompromised people, you can see the pathogenic strains of COVID start to form before you see them in the population at large, because in a immunocompromised person, that COVID doesn't have those same checks on its reproduction. So it really kind of turned that whole person into an experimental virus making factory, because the protein a virus uses to copy its genome is not great. Like it don't get me wrong, like it does a pretty good job, but like in our own cells, the penalty of being wrong in multiplying our own DNA is cancer. In a virus, the penalty for mutating your genome is potentially a more fatal virus that works better for you, the virus. So, viruses are really good at rolling those dice. And so for me, how I have shifted in my viewing is I'm no longer looking at those mildly sick animals as having developed a illness that can potentially, or develop a virus that could potentially cause illness.
Yeah.
Instead, I'm looking them more as this is a snake that is currently a virus mutation factory, and so I want it the hell out of any other rest of my animals, because the virus that, so this whole building, I don't trust for a second that a single one of these animals are negative, because again, every year I'll have a positive pop up out of nowhere. So it's the virus that is currently in my colony is not causing illness, and so I'm okay to kind of leave it at that. But now that I'm looking at these snakes, I'm looking at them as a virus mutation factory where they might roll the dice on a colony killer virus. So maybe it's I kind of wonder if we're putting, if it's a chicken or egg scenario. Like, is it the sickness that causes the killer virus, or is it the virus that causes the killer sickness?
Yeah.
Because if you kind of think of viruses as kind of part of the snake microfauna, microflora and fauna, and it's so this is what's really unique is my stance coming from cell culture, because what I do is I take a little bit of an animal, its heart, its lung, whatever, and I throw it on a plate, and I try and create the conditions for it to grow and reproduce. And with that tissue, sometimes comes bacteria that was already in the tissue, already on the snake. That it just lives there. And for cell culture, we definitely don't want it there, so we try and get rid of stuff like that. But even when we try and make the most medically scientific sterile conditions, when we're taking an in-sterile thing and putting it into our attempt at a sterile environment, we can't kid ourselves. And where I'm going with that is that, we do the absolute same thing of, we take a tiny portion of a being, the being being a wildlife population, and we take a tiny portion of that, some cells, some individual animals, and I throw them in a box with media, we throw them in a box with mulch, and we try and create the conditions for them to grow and reproduce. And the same conditions that make the cells grow also make the bacteria that grow with the cells grow, and the same conditions that make our snakes grow the bacteria that the snakes come along with. So if you think of virus as part of that, and we do know these viruses have a place in wild populations that is complicated. So it's elements of what we see maybe aren't so, I don't know where I'm going with this, but just that we're taking in-sterile beings and trying to pretend that we're keeping them in sterile conditions, and we're not.
Right.
Yeah, no, it's not.
So racks in their room, and they're like, everything's sterile, everything's clean. They're still living beings inside of your
and I mean,
room.
well, it also goes to there's a like if I'm working with a specific pathogen in people, I would order like a specific mouse that I know it's called a SSP or a SPP, specific F, man, not good at the letters there. Specific pathogen free. So it's a specific pathogen free line of mice. And this is saying that it is free of a specific type of something. Like, you can't even just say, like, this is a sterile mouse.
No.
It is sterile for one thing and one thing only.
Yeah.
So it's like the expectation of a entirely Serpento-free collection, if you don't start out with that as your square one goal, testing stuff, getting it out of the egg as soon as possible,
And
you know,
from everything
expecting a Serpentovirus-free colony just kind of might be some level of unrealistic.
Yeah.
You know,
and I
so,
think
so. And within that, I look through the filter of okay, well, clearly there are circumstances that these can kill colonies. So how do we avoid that? Because and you know, so again, fully hobbyist hat on. Here is my loosely drawn out protocol of how you, as a producer, can deal with Serpentovirus in a close to practical way. You know, you can be perfect at it. You can test every offspring multiple times. You can test every animal multiple times in your colony, and you can spend hundreds of thousands of dollars doing it. And I don't kid myself to think that that's a realistic proposition for 99% of the people out there.
Yeah.
So with that in mind, here's how I work around my colony, which is to say that this is how I work my healthy colony, where I normally, you know, I have one female blood that I said broke with that mild rest that I'm isolating. So exclusive to that one snake, this is how I would generally go about managing my healthy colony. So for starters, any for sale offspring that I plan on making available, I absolutely keep in a different room as my adults. So we don't see any evidence of vertical transmission, certainly on pythons. With boas, it might be a little more complicated because like they give birth in the cage. So yeah, but certainly with pythons, and even then, the sooner you can get them away, the better in terms of that. Like an artificial incubation is more than enough time period where risk of virus, active live virus still left on the eggshell is very minimal. Yeah. So what that means with that decent with a no vertical transmission is that even close to decent biosecurity is pretty much your best insurance policy you could take as a seller. You know, if you have your for sale offspring that you have lived nowhere else but the egg and that room, that's a pretty safe animal to put out without, you know, worrying about testing it ahead of time kind of thing. You know, I think that is certainly fits the criteria of which I would feel comfortable prior to insisting on testing before I buy, you know? So that's how I deal with for sale offspring. Specifically stuff, if I were to try and sell it and that customer wanted an explicit Serpentovirus, nidovirus free animal, I feel like the best way to do that is probably to send it with a fish heads diagnostic test to take on arrival. Because the thing is, so like my blood pythons will test negative. If I go out there and I test them, they will test negative. They don't test positive until they have a reason to test positive, and that reason is stress. They're either, you know, laying gravid because they have eggs. It's cold because they're a male and they're breeding females. You know, there's some reason.
There's some factor,
and it can even tie in with husbandry too. Like it's just again, virus is one of the many variables in the equation, and it depends so much on so many things. But yeah, so I think the safest way would be to sell an offspring with a test to take by the customer. That way the customer knows that they have taken that sample themselves. They can assure that the quality is up to their own standards because they're the ones taking it. That animal, because it's fresh out of shipment, is going to be as stressed as possible. So if there's a low-lying infection there, your chances of picking it up are better. So that's what I think is realistic. Is I actually don't think it would ever be possible to fully guarantee a truly Nido animal. And I can kind of go into some reasons why later on, but aside from that, I think that the best balance is that I think a seller can guarantee a negative result on short arrival after receipt, and I think that is probably the most realistic way for buyer and seller to navigate this if they were genuinely concerned about both protecting yourself as a seller and both as preventing pathogens from coming in as a buyer. So so that's my other pillar of how I manage biosecurity in my colony is I screen and I quarantine stuff coming in. So I go way over the top with a year-long quarantine, but that's a little probably atypical for what's probably necessary for most people. But certainly 4 to 6 months is I think pretty reasonable. And within that time span, again, if serpentivirus is something you're concerned about, which there's very good reason to be, testing 2 to 3 times in that interval is a very much good idea. So that is how you can be safe with it. So.
Yeah. And for the people who are looking at a positive test and thinking that means that their snake is going to die, that's not really always the case, right?
It's certainly not always the case. Like again, it's if you see a sick animal, if you see a sick python with a rasp, it almost always has a serpentivirus. It's like it either has a serpentivirus or we haven't found the serpentivirus because it's a little bit different and our primers aren't picking it up, kind of thing. Like these viruses are very common. So the immediate receipt of a positive result is not, don't get your thinking too far ahead of yourself. It's not the absolute end of the world. Like slow down, think about what is the context in which this virus is causing problems for your animal, and kind of work within that realm. Don't get me wrong, like I really want to make sure I'm walking a very fine line here because I really don't want to give off the impression that these viruses don't matter, and I really don't want to give off the impression to not trust testing because I absolutely think these viruses matter a whole lot, and I absolutely think testing is critical. The problem is when people have a different understanding of what they think that virus might represent, and also what question they think they're answering with their testing versus the kind of actual question they're answering with their testing.
And I think that's been one of the biggest issues whenever discussions of nidovirus or serpentiviruses ever come up is there's the people who think like, I have tested this animal 2 or 3 times and it's consistently been negative, therefore it is 100% negative. Does not have a nido. And when you throw that sidestep to them where it's like, well, that actually doesn't mean anything because as the primers change and get more intricate as you guys learn more about stuff, or if that virus mutates and then suddenly gets picked up, then you can't guarantee that that's not going to happen, or
not shedding enough sting, or you're not hitting
so you have multiple things going on here. So you have a phylogenetic tree of snakes that is big, and the viruses that infect those snakes also is looking to be big. So there's a big combination of particular type of virus plus particular type of snakes plus particular stress conditions that can certainly lead up to something that can very much wipe out colonies, but it's very tricky.
So, but there's animals who can test positive. They are kind of like asymptomatic, like the people with the coronavirus who don't show any symptoms. They're not sick. They're not coughing. No fever. None of the things you would expect to see, but they're still positive. They still carry it.
And there's a pretty good chance that that's the majority. Honestly, I would at this point feel like it's very safe to assume that it's safe to assume that the majority is an entirely asymptomatic positives is what your average serpentivirus case is. but we only find the ones that cause problems because we're only looking for
Those are
problems.
people who are testing. Yeah.
Right.
Yeah.
Right. So somebody asked in the comments, what should a seller do if somebody does purchase a snake in your scenario, sending it out with a Nido test and it ends up testing positive? What then becomes the appropriate course of action afterwards?
Yeah, so there's kind of a whole range, and it really just depends on certain circumstances. So like I'll give a specific example. So I have a group of popwind pythons, and one of the males in that group, of which I made sure to get numerous males since that's a thing you need to breed them. Yeah. So multiple males. One of those wild caught males actually tested positive for a serpentovirus, but is like totally pretty solid on health. Like, had this thing for 6 months now, and it's a solid animal. But the rest of the animals in the project are negative. So, until I need that male, I really don't want him around the rest of them. If I get to a point where that's the last male I have, then I'd roll those dice on exposing that female and knowing that, this virus in this male popwind python didn't cause illness. So maybe the same virus in my female won't. That's the risk that I choose to gamble in that scenario. And that's the risk I chose to take with the retics, because I've used my positive male retic to breed, a different female retic, and they're both, again, perfectly healthy, solid snakes. So anyway, what I did with this particular male that I didn't want hanging around the colony was I had a friend who really wanted some experience with popwinds, and they had a place. So I built a whole cage with it, and that cage and that animal are a combined unit. And I'm letting him, have fun with his popwind python. And if I ever end up needing him, I can, give him a call, and that's where my male is. But, in the meantime, I know he's not presenting any risk because he's kept in an isolated cage in a pet type environment. But I also know that if I ever do need him, because he is an animal that would very much serve a purpose in, my type colony, I know where he is. So option one is to kind of rehome it out like an FIV cat, like one of the cats with like an HIV type virus, where basically if you have a cat positive for that, you really shouldn't have another cat that's negative for it, or else they're going to kind of get each other sick. So yeah, some sort of scenario like that. And it doesn't have to be anything crazy. Even just, if you have most of your snakes in a bedroom and you want to keep your questionable one out in the living room, I think that's within pretty realm of safe if you're doing decent biosecurity. So
so it essentially all ends up becoming like situational, from the buyer
it's
to
entirely
the seller.
And
whatever
so,
you guys agree on.
So for me in my colony, I should step back and say that I have I'm sitting right now in my office and quarantine room. I also have a outbuilding where I have most of my adult blood pythons, and then I have an offspring area that's separate from all of that. And then what I have coming in eventually is going to be another like totally sterile building, which is going to be what I'm working into. But basically, so my whole building, I know what's in the colony. I know when I feel I should be scared of it. I know that because I'm not selling animals out of that colony and I have my offspring in a separate area, that those animals present relatively little risk, especially if they're testing negative on tests, because they're not even shedding enough virus to pick up as a positive. So what I would do specifically, if I had blood python that, left my facility totally healthy, ended up popping back with a positive test, and I recognized that virus as being my virus, I would probably have some trepidation. Like, I would probably put it through a quarantine period before throwing it back at the colony, but I wouldn't. If it went back to testing negative, I would probably feel some level of safe putting it in my colony. And I really need to stress that some level of safe means that I assume every single one of those snakes in that building is positive and treat them as if they are such. So, it's there's so many different ways to manage around it, and so many of them are, I don't know, you have to do some level of mental gymnastics to be excited about working with a snake, with a building of snakes that you've already, you're like already decided these are positive. You just haven't found out when yet. So, but you can essentially walk through any colony like that today, and it's essentially most likely to be the same thing. If you've done no testing and you have more than a couple of pythons and you're not seeing mass swarms of illness, but you've still seen literally any level of respiratory at anything, like, you're walking through a room with probably at least one positive snake in it, and probably a lot more than that. But
well, that's I remember.
It just
I remember when we were down at Southeast Carpet Fest, like the consensus seemed to be like, if you have more than 10 snakes, you have at least one animal that
most likely
it would test positive for nidovirus. Are those numbers still kind of appearing around the same?
It's really hard to say, because it's like, we get in diagnostic samples, but a lot of times, you can never tell if someone's sending in a diagnostic sample because it's a problem, or if they're sending in a diagnostic sample because they want to make sure they're being safe. But mass screenings of some studies that have done mass screening of adult python colonies in both Europe and US have found like double-digit prevalence in adult pythons without illness kind of stuff. So, I would be shocked if the real prevalence in pythons is less than 10%. Now, in other stuff, it's different. Like, we do have turtle serpentoviruses and lizard ones. And, if it's a reptile, at this point, we've probably found one of these viruses in it. Like, there was just a veiled chameleon virus published like last month. So,
I was gonna say, I heard some rumors about like a huge percentage of chameleons in the United States testing positive for some sort of
virus. A lot of their it was a very high prevalence in my chameleon population. So within that, but that being said, like, the risk is not represented fully between each taxa, like, but it's also kind of different in the sense that, like, we don't have very many boa viruses, but when we find a boa with a boa virus, he tends to be in not great shape. We see a ton of python viruses, and we see plenty of python viruses in pythons that are not in great shape, but we also see plenty of them that are in pythons with great shape. So, which again, I really want to make sure I'm not minimizing the risk that these viruses represent. I mean, people have lost a non-insignificant amount of, money, time, animal life, my own life effort,
sanity,
to these viruses. So, I really don't want to come here and seem like I'm minimizing this, but if the person who lost a good portion of colony of blood pythons can sit here and say that I actually still think it's probably fully possible to navigate a colony with these viruses, should mean something.
Yeah, 100%. And I think that especially as more of the research comes available and more conversations like this happen where we're able to kind of get more of an inside look, we can actually start to have those conversations that are less finger pointing and more like, let's share some knowledge about this to people who might not know and figure out how you can successfully navigate a collection regardless of the size with animals that may be testing positive.
So I have a quick demo, and I might actually have a little surprise. I just got a ding from my computer that a thing might be ready.
Ooh, I like things.
So what I did was I went ahead and ran before the program here a phylogenetic tree of all the like publicly made serpentovirus genomes. So hopefully I'll be able to pull that up and show you guys to kind of see the diverse. So these are genomes that are available online right now, you guys, Jeremy and Rob, can go to GenBank and type in the numbers that are in these and, end up with the same set of sequences I'm working with. So.
Oh, that's pretty cool.
Yeah, I'm not finding my program. Well, I'll do pause on that for now. So real quick, what I wanted to show before that, any way to rotate this around? No, I'll just make you guys like nauseous.
Okay. Yay! Look at this awesome ride.
Okay, can you guys do that to some extent?
Yes, sort of.
A little bit, yeah.
Okay, so these are all genomes that are available publicly online. So here's green tree python, blood python, retic. Here's the veiled chameleon. And we can take a look real quick at one of these viruses and see what's kind of made up. So all these like green and yellow things are proteins and kind of genes that code for proteins. But if we zoom in, we can actually see there's the genetic code AGTC, blah, blah, blah. You remember this from high school.
Wow.
Yeah, so each one of these things is a gene. So the gene that we target with our testing is this part of this second gene here, this ORF1b. Here is the S gene, that's the spike protein. So the vaccine that coronavirus, that's this gene right here, but here's it in a ball python. So what I did was I took these handful of viruses and I lined them up and compared them to each other. And so when we open this document, this thing at the top, the red and the green show how similar they are. And so you can see towards the end here, the spike proteins and all this other kind of stuff is super variable. The beginning part of this gene is super variable. The one area where it stays kind of similar is this area. And so that's where testing typically focuses in on this gene right here, because that's where it's the most consistent of all of them across the genomes.
Oh, interesting.
So when you're trying to make primers to pick up all of these viruses, so we have viruses from lizards here, we have viruses from a bunch of different pythons. So the goal is to create a primer that would pick up all of them. So you could pick them all out in one test, right?
Yeah.
But when we start zooming in, we can actually see that the actual, like, even though they're kind of similar, there's very few stretches in which case all the nucleotides are green for all of them. And so to make a primer, I need about 20 base pairs that are all the same to be able to pick up all of these viruses.
Yeah.
So this is like, what, 7 viruses of the 200 probably that we know are out there. So, yeah.
Wow.
Now this is just that ORF1b gene. And so this is that area that is very consistent between pretty much all the genomes, but is still like not that consistent. And so I'm going to show you my program here can search, like I can give it a primer and it can predict where that primer would bind. So here is primer 1. So this is primer PVTF, which is in a Laura Hoon-Hanks paper. And this is one of the diagnostic primers that I trust the most. So this is like as if it were a standard PCR. This is the forward primer. And we can see that we got it paired with the blood python, it paired with green tree, ball python, another green tree, veiled chameleon. It missed my retic and it missed the shingleback skink. But we also need a second primer so that we can have a little fragment of something, and that's our target. So here is those sequences with 2 primers. And so now the veiled chameleon, primer 1 worked, but primer 2 didn't, so we wouldn't pick that up on a test. Retic never worked for primer 1, so we wouldn't pick that up on a test. We would pick up green tree, we would pick up ball python, we would pick up green tree, we would pick up blood python, but we'd miss shingleback. So that is how a typical PCR test works. And that is like your $60 to $150 range test. There is another type of PCR called a qPCR, which is cheaper. And this test has pros and cons, but one of the cons to it is that you need a third primer. So already, just by needing 2 primers, we've lost like half of the viruses on this list. By needing a third primer to match up, and all 3 need to be able to match up, now we can only pick up green tree python, ball python, and another green tree python virus. So if I had a
Wow.
blood python serpentovirus and I submitted it for a test that was using 3 primers, the chances of it getting picked up, if that was not the virus that test was originally based on, is not great. So this really shows the difference in what we call sensitivity and specificity. So this test, which is our standard PCR, has a higher specificity. It can detect more viruses than this other test. This other test has a lower specificity, but a higher sensitivity. So it can detect virus when it's actually at a lower level. So if you know what you're looking for and you know what to be scared of, this is probably the test to choose if you know it'll pick it up. If you don't know what you're looking for, then this might be the test you're looking for, because again, you might be missing more samples just from how these tests have to operate. So yeah, that's a little on-the-fly demo of some protein virus stuff 101. And yeah, hopefully that gives you some level of sense in terms of like taking testing from this hypothetical thing of mixing chemicals in a tube to actually showing what is occurring and why something might work and why something might not, and why you might get a negative on a positive snake. Because again, if I sent in that blood python virus and it didn't match with all 3 primers, then I would have a negative thing for all I knew.
And then people go, look, I've got a Nido-free
negative,
snake. And
yeah.
it's like, well.
Nido-free snake. Or
yeah.
And I send it to someone else, and that someone else uses a different testing company that does pick it up. And now, maybe it could get so bad as that person says, well, I tested it and it's positive, and you told me it would just test negative. I bet you faked that test, So it's there's a lot of room here for a lot of hurt feelings. That I think we need to be very careful about navigating.
Yeah. Well, I think one of the things too is like, to the average person, the average reptile keeper that maybe has 10 animals, and they go through this endeavor and they get their animal tested, and you test it and it's negative, and they test it and it's positive, and then it's like freaking out. Trying to then explain something like this to them, at that point, it's so far over their head, because all they're seeing is, I have a positive animal, or this animal tested positive. You told me it was negative, whatever. So we kind of, I don't want to say we lose the ability to have that conversation, but to the average person, it's hard to fathom, these varying things that make these tests not foolproof, and it's not for obviously the lack of sciencey things that
it's really what it is all of these scientific techniques have known limitations that are just not known by anyone who's other than the people who
exactly. And I think that's the thing that is the most unfortunate about this, is like, because we get into keeping reptiles because we love keeping reptiles, and most people, again, thinking about the average person who's just getting an animal because they think it's awesome and they love having that animal, they're not necessarily thinking of anything as simple as, well, what's the scientific name of this snake, let alone, hey, let me understand how PCR tests work.
Yeah.
Right.
What I mean? So to then have those conversations, or have to have those conversations, it takes a little bit, I don't want to say it takes the magic out of keeping reptiles, but it really makes you want to understand, like, okay,
I will fully
science
vouch for it
this.
taking it fully takes away the magic of the animals.
Listen, really, Steve, you're a doctor. You're a doctor.
No, but that's the thing.
Everything's horrifying to you.
No, see, the thing is, I don't I didn't set out in life wanting to study viruses. I studied viruses because I had a tremendous hole in my life created by a virus that I wanted to figure out how the hell that happened. So and what is interesting and where I see myself as fortunate, kind of before my time in some sense, is that I learned the lesson that you can build the best system in the world, and all it takes to topple it sometimes is 30,000 nucleotide bases. And lo and behold, 2 years later, the world learns that exact same lesson, that you can build the best system in the world, and all it takes is one little thing, and we can topple it, right on over. So the fear is warranted, but also breathe. You can probably still put yourself in the position of being unlucky if you lose animals to serpentovirus, but you can still apply unlucky at the population scale of herpetoculture in the US and end up with a lot of unlucky people. And it's hard to parse and navigate your way through those two realities, because you can do absolutely everything right and still end up in a place where you didn't think you'd be.
Yeah, for sure, man. There's certainly so much to discuss and discover as obviously as the science is still unfolding, because it's still
learning.
It's still learning. Yeah, things are still in the very new stages.
So I finally got my tree up, so I'll show it real quick, and this will be the last we can talk about this, and then we can talk about fun snake stuff and be done talking about all this stuff. So so this is the phylogenetic tree that I just ran. It just finished on air. This is the first time I'm looking at it. There's actually some samples in here that I'm really excited about, including that popwin python virus. So now I finally get to reveal on air where he fits in the phylogenetic tree of serpentoviruses. So let me take you for a ride again. All right, can you guys see that?
Yes, it's very, very hard to see, but we can see it.
It should be very tiny. Actually, hold on. I think I can increase the size.
There we go.
Yeah.
Look at that.
There we go. So these are the numbers that if you search them up, you will find some of these samples. Some of them are not published or don't have those numbers, but they're samples from my stuff, so I don't feel fine showing them. So these are the ball python viruses, and I would definitely say that by and large, when we see problems, there is oftentimes ball python or a virus very similar to ball python sitting there as the problem child. So I would basically say that like anything in this plate is kind of scary business.
Mm-hmm.
Over here, we have a bunch of green trees and my blood python virus that, again, I know in some contexts can do nothing, in some contexts is scary. And then here is that new popwin virus. So I actually did think he was going to sit alone by himself, and he's not that really closely related to anything. So it's he's kind of different, kind of weird. And here's my retic. So he's like way off on his own.
Really?
So it just goes to show that there's huge diversity here. Here's shingleback skink. So this was found in a skink, failed chameleon. This was found in a water snake nematode. This was found in I think a wild like mandarin rat in China.
Wow.
So like these viruses are out there, and we definitely see they break down. Like these are colubrid viruses, but they don't fully, like these are mostly python viruses, but then you have a weird lizard virus in here and a lizard virus. So it's just nothing. Oh, here's the emerald tree boa virus. Yeah, here's the emerald tree boa virus is most closely related to cow nidovirus. So it's
what?
Yeah, genes are all over the place. So viruses are weird. Viruses are crazy. Family's really big. Expect a lot of virus diversity. And when you're talking about Nido or Serpento, like this is what you are cumulatively talking about. And this is just the publicly available full genomes, because we have like hundreds of other samples that are also published that just have partial genomes. So it's like,
wow.
This is like getting a rough shape of the general shape of how big the iceberg might be. This is not, I can't even go so far as to say that this is the tip.
Damn, this is less than the tip.
That's wild.
Yeah. So there you go. Last snake talk, that was everything. I made an agenda ahead of time of info I really wanted to hope to be able to show people, and that was the last bit. So now we can talk about cool snake stuff and projects and all that jazz. Let's talk about, we talked about nothing but viruses and a dead ball python. So let's
the immortal ball python.
The immortal ball python. That's true.
So what kind of project, what kind of stuff you got going on behind you there? Because you do have some. You said that this is your quarantine room.
Yes, this is the quarantine room, and this is also where I get to do all my sort of fun experimentation stuff.
Oh, is this the rack?
Yes.
Yeah, this is my work rack.
Yes.
We see a scrub python just chilling there, sitting in the fork of the tree that I'm able to provide through this type of caging. And what's really cool is that I can like fully remove this bottom section and do any sort of maintenance I need without, fully disturbing him. Although, I did, but but yeah,
They're
I've
smart. They know what's going on.
really taken it upon myself to really experiment with just some weird, wacky new ways of keeping and new ways of building stuff. You can see I kind of have like some weird. I think she has one. Oh, she just knocked it down. Like elevated perches and stuff like that. So it's it got really playing around with a 3D printer and all that jazz. And one thing kind of led to another. I realized that I could do this, and then I could do that, and then I could do this other thing, and then I can. So anyway, where I'm at with it now, because I built those like modified weird Freedom Breeder racks about a year ago. And I'm actually to the point where I'm like, screw the tubs. Like half of the challenges in that design was making it so that it worked as both a rack and a cage.
Yeah.
And then I realized that the benefit the rack provided was almost nil. What I really want is a cage that slides out like a rack.
Yep.
And so I already have that. Like I can pull out that top section like a normal cage / tub. Like it opens like a tub, works like a cage. And I figured that's what I wanted. Why the heck am I spending so much effort creatively trying to accommodate this bottom tub and also wasting 2 levels of a rack when I could just, have a suspended cage off a Freedom Breeder frame, which I think is, I think that would look sharp. Ooh, I think that would look good. Yeah. So that's probably the direction I'm heading it in is that, I have a bunch of Freedom Breeder equipment and I love Freedom Breeder, but, I've hit the limit of their equipment, of what their equipment can provide. And, I want to try something new. So, that's what I'm moving towards in the future. And also, I like seeing this stuff. I didn't realize how much we were missing, even in a clear tub.
Yeah.
Like it's we don't know anything about these animals that we think we do. It's like the PCR. We're asking one question, thinking we're answering another.
Yeah.
Yeah.
Yup.
For sure.
Yeah. And dude, I love the seeing the updates you've been posting of like the pop ones and then the scrubs and everything. And I'm just like, oh yes, I just more.
Oh yeah. So long term for my colony, what I've been doing is trying to get some of these very kind of obscure python species and just doing these incredibly over the top year-long quarantines, testing the snot out of them. And I said I have another clean building coming, and that's going to be like, an entirely biosecure thing. And so the goal is to have I'm worried about pythons. I'm worried about these viruses. I'm nervous that they're out there. And I would feel a good sense of reassurance if I knew that there was some place where there was definitely pythons and definitely not virus. And I figured I of anyone would be the type of sort to be able to create such a thing. So that's what I'm working on.
Yeah. I think that one of the things that people don't think about, like when people think about, oh, well, I brought a snake into my, into my collection and I quarantined it and blah, blah, blah. The thing that I think about is how did that snake get into the US? Because like with the scrubs and a lot of the imported stuff like pop ones and stuff, the people who are collecting them are probably taking 30 of them and putting them all in a bag and then carrying them to wherever they're selling them to, whatever port or, wherever skinning center that they're bringing them to. And they're just carrying a whole buttload of these things together. And you got to figure that if there's one infected animal in there, then they're all
well, and we got to think too that so what we do here is we've changed the entire like mathematics of what a virus will operate under in our captive conditions by creating these very dense populations we actually changed the type of trait that would be selected for in a virus to do well in that set of circumstances.
Yeah.
So again, it's like we can pair these kind of probably pretty inert background viruses that they carry from the wild, and yet place them under conditions which we can suddenly all of a sudden select for inadvertently the type of thing that's going to spread across the shortest area possible in the fastest time possible. And again, you can make I guess it's we don't know what makes a killer virus.
Yeah.
But we also know that there are plenty of not killer viruses.
Yeah.
So is there any species that you've gotten that you're just like, I wasn't expecting to like these as much as I do?
You know what is so in this room I have sabus, water pythons, Timors, polyretics, some ring pythons. So
the
the
ring pythons.
I love ring pythons, but I know that.
So I actually have some. Yeah, they are, and I got some really weird ones. Like I was like, if I'm getting into ring python, so I have some like really nice ones out in the building that are like your classic banded high contrast ones.
Mm-hmm.
And
like
so what I did with these two is I'm going to pick the ugliest ringed pythons I can possibly find and just go as far left as anyone else as I can. Because again, my goal is to create an isolated closed colony of pythons of obscure species. So I wanted the maximum divergence of phenotype to really kind of get that full spectrum. Hopefully you can use that as a vehicle for other genetic variation, but probably not really. But anyway, I have some really weird ring pythons that ended up actually turning like really cool, almost at a very smallish stage. They're looking like almost jet black. They're unfortunately both in shed.
Literally what I was gonna say, because literally people bullshit and talk crap about Bismarck ring pythons. They're like, well, they're not orange when they are adults.
God,
look,
God, what is that? Shit.
And here's the deal: people love all black fucking white lip pythons. They're like, oh my god, it's the prettiest snake on the planet. And it's like a Bismarck ring python is almost that. And if you had an all black one like that, it's even cooler. Oh, that's awesome.
Exactly. But actually, so I would say though, out of all of the species, the weirdest to me has been the Timors.
I was just gonna say that. That.
They just so you have two species in my mind that are just weird pythons. Like they clearly don't fit in any other box of any other python, which is the Papuan, which is kind of intuitive because aesthetically everyone's like, oh, it's just an olive python. Like, but like if you spend 5 minutes with one, you're like, this is I've never worked with any other python like this.
Yeah.
The second species that's weird like that is the Timor python, because it just has all these weird elements that it shouldn't have thrown together in one place. So weird element one is the genetics of a retic, is Malayo python, and like very little about Timor pythons trigger any of the retic bells in my brain.
Yeah.
So that's weird thing number two. Weird thing two is that they have like a weird liasis tail. Like they have this stubby little liasis tail that like again, it's like that is many things, but it is not a reticulated python tail. The head scalation, they have these giant head scales, not a retic.
More like a
trait.
Yep.
More like, and then the weirdest thing to me is, so the only other pythons that I can think of that have a transitioning pattern, which is to say that like there's some snakes that have like variation in their pattern, but like the only snakes that I can think of that start as one pattern, their middle is a different pattern, and their end is another pattern are scrub pythons and for some reason Timors. Like they should not be in that bucket. Like even a barneck scrub and a like southern scrub don't have the same transitioning pattern in the same extreme as a Timor and a barneck scrub. Like it just they're goofy pythons. They shouldn't be. They just shouldn't. It just
Oh man.
See, I look at Timors and like I've liked them for a long time, but I never felt that like push to get them. And then now I've been seeing more of them, and I'm like, oh man, they're just so cool. They're so interesting to me.
Yeah, I can't say that they're like necessarily my favorite captives, They definitely like to pee a lot, but they're not bitey. So like, I'm sure you can be habituated down with it, but like they explicitly just really like to pee for some reason, and that's like, and it's just off-putting. But aside from that, they're otherwise seem relatively decent. But the same thing, like they've always been a cool python, but I never felt like the sudden urge to it. So much as just the fact that like if I want representation of Pythonidae, this is clearly a python that wants representation.
Yeah.
And then once I got him, I'm like, oh, you guys are weird.
I love it.
Hell yeah.
Damn. I'm still geeking about that all black ring python.
Thing's awesome.
I really like ring pythons, so that to me is like, what the hell? Yeah.
Yeah, they've not been a fun project for me, but
I know that from like a lot of people who keep them. They're like, they're not really great. They piss all over. They bite like a motherfucker.
They really like to eat each other or try to eat each other, and then it's one of these days I'll breed them, but man, it has not been. I've not had great luck on the project though.
Have you talked to Chris at Mystic?
No, I haven't. Really, I think my problem is I have 2 females, which is why I got an entire redundant pair because I'm like, so I had at this point. By the time I had suspected they're 2 females, they're like 2 adult animals to the point of like sexing them is like gonna not be fun.
It'll hurt.
So I'm like, So I'm like, I'll just buy a neonate pair, and then I know I have a male, and I also have an ultrasound. And I felt like I definitely found follicles in both of them at some point. I don't remember. I have too much going on in my head.
Being a doctor and all.
Yeah. At any rate, so now I have something.
Come down there next time. We should sex them.
I'm down.
Really, my thing is that I know I have a male now, so I'll just see which one gives me eggs and doesn't get eaten. But
cool. Doesn't need a blood donation.
Yeah. That's amazing.
Oh, here's another random thing that I am excited about that is a plug for a company that is not giving me anything to drop them, but so like. I'm really excited. Husbandry Pro is like a new data software that's come out. And okay, so I'm like Mr. Data. At every podcast I've ever been on, I've talked about at length how important keeping data is. Yes. And then inevitably, you or whoever it is ask me, okay, well, what should your general keeper at home use? And then I'm like, crickets. Like, there's no good options out there. And I'm really excited because it looks like Husbandry Pro, they're serious about it. Like I'm not gonna lie, like I went in day one and like really wanted to see their system put through its paces, and had a list of stuff that I would like to see if I were to switch over from my own database. And they've done a very good job at hitting those items like in a way that has truly impressed me, to the point of where I'm able to vouch for a final queryable data system for tracking and keeping animals that I think actually has enough flexibility to be both useful and it's both. It provides a good balance of ease of use and utilitarian output from it, like in the sense of you don't have to change your workflow to fit the system into it. I feel like they've done a good job at that and made a good job of extracting meaningful data from that. So that is high praise that I am very relieved that I finally get the chance to, like, for anyone who's ever asked me what they should use, that is where I'm gonna point you towards now. Because because again, it's good enough where like I was at the point where I would have to make a better front end for my database to do everything I wanted to do. And then it became apparent that I think their system's going to be able to do everything it needed to. So why bother?
I was to say, Billy from Mutation Creation just adopted that, and he's really enjoying it. He's been promoting it too.
He has been promoting it crazy. Yeah.
Like I said, because I've done enough of these database stuff, like the type of suggestions I sent them were like not easy suggestions, and they kind of cranked out at a rate that would like make me feel pitiful. Like they're to a degree that it was very clear that they're very serious about it, and that gives me like good hope that there's actually some innovation coming in there that I'm really excited to see, because snake keeping with data at scale is, you guys know me well enough to know that checks all my boxes.
Yeah, for sure.
That's awesome.
If it makes Steve happy, that means it's good. Oh my gosh.
It's like how, when I'm like, I give it the thumbs up, you're like, oh, this must be
good.
good.
This is
Rob,
good.
Rob doesn't give
if
it.
You got the thumbs up from Rob. That's you're gonna want to check that out.
Probably gonna be critical of it.
Oh my gosh, dude, it's freaking awesome.
Hell yeah,
it's freaking awesome. All right, I think it's about that time.
It is about that time. We've run a little long.
Yeah, we've run a little long, but it's okay because we got some really great information.
And this was a much needed episode.
100%.
Yeah.
100%.
And I guess I can also, if you guys have any sort of supplemental material to hand out, I can probably put in screenshots as well to make this a little more accessible to people who didn't, aren't looking at a video feed or some of it.
Yes.
Instagram.
What?
For the Instagram, that if he's got stuff that he can screenshot.
Oh yeah.
Oh yeah, that
sure.
Boom.
Yeah, if you and any links that that you have to stuff that we can send people to, just text them to me and I can put them in the description of the video and the audio stuff. So if people want to check out more about any of this genome stuff, sciencey things from the doctor. But yeah, you can text me those and I'll make sure that they go up in the video descriptions and everything like that, so people can check it out.
All right, Steve. So I'm gonna ask you. I'm gonna throw it right at you. The question we ask everybody: what right now in the realm of reptiles got you excited? What is it? What's got you geeked? What's got you stirring?
So there's an energy right now, and I'm really excited and curious to see where it goes.
Mm-hmm.
We got a lot going on in the world right now. There's a lot of pent up something.
Yes.
And we're coming to a point where the uncorking of that is, approaching realistic.
It's happening. Yeah.
Yeah. So that has me excited. I'm really excited to see how the money flows this year, if it flows in the same way as it did last year, which was unique and weird. I'm curious to see how reptile expos go. I'm really curious about all of it. I'm curious, I could see everyone just roaring 20s and sitting at home. They want to blow out to their reptile expo and send a bunch of money, or I could see them wanting to go and spend their $1,500 in Cancun because that's finally, so
it's an option.
I'm really curious to see how how that actually shakes out. But kind of building on that and that kind of energy is, I really do feel like there is a hunger out there and it's growing for anything different.
Mm-hmm.
And I don't just mean different species wise, because that's certainly where I think it is most easily felt, with the people drawn towards weird, obscure species now becoming back in fashion and seeming of having value again and stuff like that. But I also think it's hunger and a new, not a new type of keeping, but just a new, I don't know. I feel like people are starting to view their animals through a slightly different lens, and I'm really excited to see where that goes. And I think a lot of that has been wrapped into people opening up to other species, but I think there's some other elements outside of it too so yeah, there's not a great answer to your question because it's sort of a je ne sais quoi of an answer, but I'm really excited to see how the year goes, because I have no clue how it's going to go.
Yeah,
because there's so many different positive and negative scenarios that I could see work out. Certainly any scenario which doesn't involve a global pandemic is certainly a net positive, but
yeah, for
get
sure.
what I mean. But so I'm really excited to kind of see what shakes from that, because I don't know. There's a new feel. Different people asking different questions. People getting excited over stuff in ways that feels new.
Mm-hmm.
And I'm really excited to see how that gets translated back out when the real world is open again. How much of that sticks and how much of that stays. And I think I think that's the same type of questions everyone's asking in every element of their life right now. And it's gonna be really neat.
Hell yeah.
Hell yeah, dude.
I'm here for it, man.
I dig it.
That's awesome.
I dig it. All right, if people want to find more
He doesn't
about
want them to find.
No, wait. I have a place to point them this time.
Yes.
Ah, there we
yeah.
go.
Yes. Okay. So
All right,
people
where do
want
we
to find out more
send
about
them
you.
to?
Where do we send them? Where are they gonna go?
So I have been trying to reinvigorate my YouTube channel again.
Yes.
Yeah, because I clearly I have a lot of kind of neat projects going on in my life that I feel like is very different than what a lot of other people are doing, and you know, I've been waiting around for someone else to do it, so I guess at this point I just need to talk about the cool shit I want to talk about.
Go
Yeah.
get the doc on
So-
YouTube.
Yes.
So the problem I have is that I don't have any time for editing, so I might have to figure out some logistics on that. But in the meantime, I will point everyone towards my YouTube channel, which is Reptillis, REPTILLIS. I already have some pretty neat videos on there so far. So if you're not subscribed to that, go subscribe to that, because I'm gonna have more cool stuff. And I think it'll be, again, weird pitch for myself. Different. I think it'll be different.
That's right.
That's right.
That's right. I
Don't
dig
read
it.
the comment section.
Hell
Don't
yeah.
read the comment section.
Just don't read the comment section.
Yeah.
Actually, you know what? I'm gonna revise that, because the people who are listening to this right now will probably leave useful feedback, because I've gotten a lot of really good feedback on my YouTube videos.
That's fair.
And the last time that I had a foray in YouTube, I did not get that. So it's been nice.
Yeah.
Yeah, I mean, the whole reason I shut down my channel in the first place is because, waking up to an inbox of a dozen emails telling me how ugly my hair is, and how stupid my voice. Like, I was like, this is not fun anymore at all. So yeah.
Hell yeah.
So I'm hoping that goes somewhere, does something cool, because again, it's I feel like I have a tendency to approach things in a slightly different way than other people, and maybe putting a little bit of that out there more might get other people thinking out of the box as well.
Hell yeah.
Yeah, for sure, dude.
All right, man. Thank you so much for coming on tonight, man. It has been amazing
dude.
chatting, and I have had a really good time listening to the info that you've been able to share with us tonight.
Yeah, for sure, man. Thank you very much.
Yep, it's been great as always. Thank you for having me.
Hell yeah.
Yeah, man. We'll have you on again soon.
Yes, and I
Yep,
actually
absolutely.
mean soon.
Yeah, yes.
All right.
All right,
Take
dude.
care, man. Have a good night.
See you,
Later.
man.
Talk to you soon. Boom.
Boom. Another one down. Steve, the doc.
Yeah.
I'm gonna start calling him
stuck
that.
around for this whole episode. I applaud you, because
Yes.
it's not always easy for people to understand. Even if you have a background in science, some of this stuff is very complex.
Yeah, for sure. So if you want to learn more about anything that Steve is talking about, as soon as he gets me links, I'll make sure that they go in the video description. His YouTube handle is already in the description, and it'll be in the description of the audio only. So go check that out. Subscribe to his channel. Go show him some support. And of course, you can find us on Instagram at reptile.talk, at Rob is Creeping It Real,
At Brassman Reptiles.
and yeah, do that. Do
Thank you
more
so
of that.
much.
Thank y'all for hanging out.
And make sure that if you are listening on Apple Podcasts, leave us a review.
Yes.
Let us know what you think of the podcast so far. I've noticed that the audio quality has been a lot better since we hopped on StreamYard
Yes.
and got things swapped up a little bit here. If you have not, share at least one episode with somebody. If you shoot a screenshot over to someone, to me, a screenshot of you sharing with somebody,
Oh,
we've
that's
got
right.
that
That
contest,
giveaway. That's right.
where I'll be giving away a temp gun at the end of March. So we got,
That's
I think,
right.
one more episode, or it might be this episode is the end one. So we'll be
I think
picking
it might
at
be
the
this
end
one,
of
yeah.
March. We'll be picking a winner, and we'll shoot you out a really nice temp gun, because everyone should have one in their reptile room. So thank you guys so much
That's
for
right.
checking it out. We'll see you next time.
See ya.